Hey guys, let's say you are investigating the effect of a treatment for obesity for 20 patients. From the patient sample, you obtain thousands of transcriptomic data. How can we make sense of the data? As we know, most of the data might be irrelevant. Can we focus on the relationship between only a few groups of genes and obesity? In genomic study, when we have a bunch of gene expression data, the easiest way to make sense of the data is to form a correlation among the trends of the expression set. For example, gene A and B are having similar functional annotation. Let's say they are both the genes in the insulin pathway. So they are normally grouped together, and then we investigate how many genes are upregulated and how many of them are downregulated. So in the treatment group, if the gene exhibits an opposite trend compared with the other genes, it suggests that the gene plays a role in changing the condition of the cells and might be the targeted therapeutic gene. Wait, while it seems logical to do this way, it will lead to a highly subjective conclusion. What if the treatment does not affect the targeted pathway? What if the biological function of the gene leads us to a wrong assumption? And also, it is quite tedious to look into each particular pathway in detail in order to find out the correct gene marker based on the differential expression analysis. Our analysis could be better. Therefore, here comes to the good use of WGCNA, weighted gene co-expression network analysis, a statistical workflow to form an association network for the gene expression sets. WGCNA tries not to predict the relationship of the genes by simply calculating the correlation of the expression profile. Instead, it does include the correlation calculation step, adding with another algorithm to measure the similarity of the genes. The output is a much confident clustering of genes, which they name it as model. Let's take a look at the concept of WGCNA. In WGCNA, the relationship between genes is better described using a Skillfree network. So what is the Skillfree network? When we simply connect the ejection dots, it is called a random network. An example is how we plan the highway route around the cities or states. Then, a Skillfree network is having a central point connecting to certain nearby dots, and these central points will directly or indirectly connect to the other central points. A real-life example is our airline route. When it comes to the biological field in genomic study, it is well fit a Skillfree network. As we can see from the graph, most nodes have only very few connections, while very little nodes have many connections. Those are called as hub genes. The number of links per node follows a power law distribution. Genes always appear as indirect correlations. Let's take a look on a biological pathway. We can easily observe a Skillfree network in most of the pathway. As seen from the pathway map here from CADE database, which is about cocaine addiction. Our biological dysfunction, like the brain, are complex hierarchical networks, and many diseases or disorders often occur as failure in network regulation. Therefore, understanding networks provides us a clue of what happened to the interaction of genes. I hope by far you understand what WGCNA is trying to do. And now, let's dive in deeper into the workflow and algorithm of WGCNA. It involves a little bit of jargonized terms, but it is not hard to understand. There are 5 steps in constructing WGCNA. First, construct a gene co-expression network. Second, it identifies models. Third, relate models to external traits. And the fourth and the fifth that involve the downstream analysis, which is study module relationship and to find the key driver in the interested models. To be able to construct a gene co-expression network, we have to able to calculate the relationship between two genes. So first, we have to calculate similarity metric among genes, which is the value range from -1 to 1, and this is similar way to calculate correlationship using the statistical method such as the Spearman method. Then the similarity matrix will be transformed into an adjacency matrix. This is to mathematically to transform the matrix into a skill-free network. There are three types of network for us to choose. First, unsigned. Genes are similar if they are strongly correlated, that regardless of the direction of the association. Second, the signed network. This is only applied to genes that are similar if they are positively correlated. Those genes that have an opposite trend are considered not similar. Signed hybrid. Only those negative associations are all treated as zero in this matrix. After we choose the type of the network, next stop threshold beta are evaluated to determine the most appropriate beta to transform the similarity matrix into a skill-free network. So, to mimic a skill-free network, there is a formula to calculate how to get a skill-free network mathematically. So, there are two ways to decide. First, to the skill independence, and the second, mean connectivity graph. For skill independent graph, we look at the R-square for how well it fits a skill-free network. So a higher value means it is closer to the skill-free network. Mean connectivity graph means we look for the minimum numbers that connect to the others, as in skill-free network. Most of the nodes are with minimal connectivity. By choosing a correct soft threshold value, WGCNA defines a correlation matrix up to a power beta, so the degree distribution will fit a small world skill-free network. Once a soft threshold is determined, we can calculate the adjacent similarity by using formula. And now we have to create topological overlap matrix (TOM) from adjacency matrix. This is an interesting part. The purpose of this step is to incorporate both direct and indirect relationships into TOM. So this is to confidently calculate the relationship of two genes by measuring their similarity. Let's briefly study the formula. Wij is the term of gene I and gene J. Aij can be easily obtained from the previous step, which is the adjacency of gene I and gene J. And then Lij measure the indirect relationship between gene I and gene J. So this is done by multiplying the adjacency of gene I with the other genes and gene J with the other genes, and then adding them all together. So the bigger of this value, the more they are with overlapping indirect genes. So the bottom part is the scaling factor, and then it is easy to calculate the next step, which is dissimilarity among genes. And now we will have a nice metric to represent the relationship of the genes. So we can go ahead to identify modules. This can be done by forming the hierarchical clustering and then the them using the metric value, comparing them pairwise. WGCNA has its own sophisticated way to calculate the threshold cutting points to divide the cluster, which is called dynamic tree cut. This algorithm allows us to identify nested clusters, and user can flexibly adjust the cluster parameters too. With this, we can get better detection of outlier too. Anyway, this step involves a slightly complicated mathematical model. If you are interested to know more, you may try to read the paper that I will put a link in the description box below. So, the next step is to relate models to external information such as clinical data, like cholesterol level, phenotypic data, or some metabolic data. In fact, this part is very similar to how we connect the gene expression data with other behavior or phenotypic data in survival analysis or the common differential expression analysis. The reason to assign the connection to the external traits is to form module agent gene. It is used to summarize the expression pattern of the module genes across samples, and this is similar to how we want to focus on a certain cluster for any downstream analysis. So, for example, from this graph, we can see that the models are associated positively with back. Just to bear in mind that to make this work, we have to make it a signed network, which is to look for the positive association earlier. And this step is considered giving us a whole picture to see the pattern or significant gene groups, to do enrichment analysis, and to make sense of the data comparison. Lastly, find interesting genes for the future study. There are two that we can further study: intramodular connectivity, whether you want to get a target gene from the hub genes or the indirect genes that in the similar module. Second, assessment of module cohesion related to an individual gene, to identify which gene is responsible for the real causal relationship of certain disease or disorder or behavior. And of course, there are many more downstream analyses we could do to answer a wide range of biological research questions. Before come to the end of the introduction, we should aware that WGCNA has its limitations. WGCNA can only assume linear or monotonic relationship among genes. Skillfree networks may be suitable for protein-coding genes, but since we know very little of the rest of the DNA, how does it fit towards non-coding transcript or other omic data? Also, WGCNA makes a hypothesis in a way to assume that co-expression of genes is kind of ignoring the possibility of being linkage disequilibrium. Robustness or repeatability is highly dependent on the sample size and quality of the data. Therefore, WGCNA has some preset criteria in order to get more robust and refined in results, such as the number of the sample has to be about 15 or more than 20 samples. The data has to be carefully collected technically or biologically. You may find out more from the official tutorial website in the FAQ section. I will put the link below as well. In summary, WGCNA analyzes high-dimensional data across multiple samples by forming a Skillfree network and assign the genes to different models, and from there, we can identify candidate genes that may be associated with the condition. That's all for today's topics. I hope you have learned something from this video. Thanks for watching, and I will see you next time.